Our topic for today is the detoxification of arsenic in mine tailings. Dr. Yongbin Lee is affiliated with the National Regional Joint Engineering Research Center for Soil Pollution Control and Remediation in South China. One of his most significant research contributions is the article Sriracha spp are responsible for nitrogen fixation fueled by As3 oxidation, a novel biogeochemical process identified in mine tailings. The central hypothesis of this article is that as dependent biological nitrogen fixation is likely to occur in mine tailings and other environments that are rich in arsenic 3 but poor in bioavailable nitrogen. The study had four clearly defined objectives. 1. Examine the occurrence of chemolithotrophic N fixation fueled by As3 oxidation. 2. Investigate whether as-dependent biological nitrogen fixation is widespread in different oligotrophic habitats. 3. Identify bacterial taxa responsible for as-dependent biological nitrogen fixation. And 4. Predict the metabolic potential of the responsible bacteria. In the microbial community, chemolithoautotrophic bacteria in mine tailings oxidize arsenite as an energy source. In this process, arsenite is converted to arsenate, releasing electrons that enter the bacterial electron transport chain and generate a proton motive force for ATP production. A specific strain of Serratia in the community uses the ATP and reducing power generated from arsenite oxidation to fix atmospheric nitrogen. The biological nitrogen fixation reaction is the following. This equation describes a process where atmospheric nitrogen is being converted into ammonia, and the final products are arsenate, a less toxic, more stable form of arsenic, and ammonium, bioavailable nitrogen that supports microbial growth. This investigation was conducted in a tailing zone of the Ziguangshan mining area in southwest China, where samples showed high concentrations of antimony and arsenic. Two different treatments were established to investigate the occurrence of arsenic dependent biological nitrogen fixation. One treatment included the addition of arsenic 3, while the other did not. In Figure 1a, we can see the substantial increase in nitrogenase activity in the AS3A amended cultures relative to the controls. In Figure 1b demonstrates that after 35 days of incubation, the delta 15 nitrogen values were significantly higher in cultures amended with As3 as a confirmation in Figure 1c about 2 millimoles per liter. As3 was completely converted to As5 after 7 days of incubation in the treatment, where As3 was added. DNA stable isotope probing was used to identify bacteria responsible for arsenic dependent biological nitrogen fixation. The abundance of the nitrogenase gene, NIFH, a marker for nitrogen fixing bacteria, was quantified in each fraction at 3 time points. Days 21, 28, and 35 shows in Figure 3a. As shown in Figure 3b in the 14 NAs treatment, the highest relative abundance of the NEFH gene was found in the light fractions at day 21. Importantly, in the treatment with 15N2 but without As3, no significant shift of the NIFH gene to heavy fractions was detected. This result indicates that the presence of arsenite was essential for active 15N incorporation by the diazotrophs, confirming that arsenic-dependent BNF was occurring under these conditions. In Figure 4a, we can see the selection of DNA fractions with the highest relative abundance of the NIFH gene from the 15NAS and 14NAS treatments at days 28 and 35. In Figure 4b, we can see that bacteria belonging to the genus Serratia dominated the heavy DNA fractions in the 15NAs treatments, with an average relative abundance of 25% at day 28 and 56% at day 35. This suggests that Cerathea species may play a key role in arsenic oxidation driven biological nitrogen fixation. Environmental implications. In mine tailings, high arsenic levels and limited organic carbon restrict traditional heterotrophic nitrogen fixation. Under such conditions, arsenic dependent chemolithotrophic nitrogen fixation may become a crucial alternative for producing bioavailable nitrogen. This process supports microbial life and nutrient cycling in these challenging, nutrient poor environments. That was all for today. See you in another research video.